We will start the molecular markers module by first discussing one important consideration when you're trying to figure out what type of marker to use. So before we even get into the types of markers, which we'll discuss later in the module, first we're going to discuss different general categories of markers in terms of whether they're neutral or adaptive. So before I explain to you, I want you to think first based on your reading or just based on what you can guess from the names, what a neutral marker is and then how that might be different from an adaptive marker. So I will stop talking and let you think about that on your own. So a neutral marker is one that does not directly affect an organism's fitness. So neutral generally means having no effect. So in this case, what that means, it has no effect on an organism's fitness. An example of a marker that would be neutral is something like a microsatellite. We'll talk more about this later in the module, but overall, a microsatellite, as is shown here, is this stretch of repeating nucleotides. So at locus 2 here, the particular microsatellite is the 3 nucleotide sequence TAA repeated many times. And so if you think about it, that makes sense that this marker would be neutral, because if you have in this organism a bunch of repeats of this nucleotide sequence, and in fact you can have different numbers of repeats for different alleles, it would be actually harmful to the organism if you were having these big changes in terms of the number of nucleotides that are there at a gene that actually had an effect on fitness. So a microsatellite is one example of a neutral marker that does not affect an organism's fitness. So here's a true-false question for us to think about this. A neutral marker can tell us about adaptation to environmental conditions. Do you think that is true or false? If you think the answer is false, you are correct. And that's because, as we said before, neutral markers don't affect fitness. So if they don't affect fitness, they can't affect how an organism adapts to its environment. That's essentially another way to say how fit they are. So a neutral marker cannot tell us about adaptation. So that's important to think about if you are designing a study where you want to know about how that organism is adapting to its environment. You don't want to use a neutral marker for that. In contrast, adaptive markers are these functional genes that influence phenotype. So phenotype, remember, are these more observable characteristics. So it might be something like an organism's growth or some kind of physical appearance, its color or morphology, something like that. So adaptive markers are functional genes that influence phenotype and therefore directly influence the organism's fitness. So an example that's cited in your textbook of an adaptive marker is the major histocompatibility complex gene, or that's often abbreviated MHC. And that's what's shown here. The MHC gene is really important in our immune system. So MHC molecules interact with T cells, and they're really an important integral part of activating our immune responses to different things that we might be battling in our environment. So because of that, because our immune system is definitely tied into our fitness, an MHC marker would be an example of an adaptive marker. Here's another true-false question for us to think about. This statement says, natural selection influences the distribution of both neutral and adaptive markers. Do you think that statement is true or false? So the answer is false. And if we think about, we just said that adaptive markers affect fitness, neutral markers do not. And natural selection is a process that's acting on an organism's phenotype to increase its fitness. And so because of that, natural selection influences the distribution of adaptive markers. That's true, but natural selection does not influence the distribution of neutral markers. Although I do have to mention, of course, with everything, there's exceptions and complications, right? So the caveat to that is something called linkage disequilibrium. And so I'll explain what this means. But what we have in the bullet point here is that neutral markers can look like they're non-neutral if they are linked to adaptive markers. So what do I mean by linked? Linked means if you think about a chromosome has several different genes located on that chromosome, right? And so if you have two different genes, this is gene 1 and this is gene 2, if they are physically located close together on the same chromosome, then they can be considered linked, meaning they're going to follow the same types of evolutionary patterns just because they're physically close to each other on that chromosome. So that's what this term linkage disequilibrium means. It means this is something that happens when you have two genes linked close to each other, they're physically located close together. Because of that, they are independent, operating essentially as the same unit. They are not operating independently. And so that's what's shown in this cartoon image here at the bottom. So say we have each of these lines is a different person. On the left, we have what their chromosomes look like before selection. This region here in red, this is actually an adaptive marker. So just like we said, natural selection affects adaptive markers. So what would happen is after selection happens, we see that adaptive marker is much more common in the population. Before it was only in one person, now it's in 1, 2, 3, 4, 5, 6, 7, 8. So a large proportion of the population now has this adaptive marker. But if we look, a large portion of this population now also has this other marker that is located right next to the adaptive marker. So everything in blue here is neutral, and like we said, the red one is adaptive. So what happens with linkage disequilibrium is that because natural selection is acting on this red adaptive region of the genome to increase its frequency because it helps the organism be fit and survive its environment, because those two genes are located physically close to each other, that neutral gene is also going to become prevalent in the population. So that's what we mean when we say neutral markers can appear to be non-neutral if they're linked to adaptive markers. So if we didn't know that this red locus was an adaptive gene and the blue one was neutral, we might think that both of them were adaptive because they are increasing in frequency in the population, but that's really only because of this linkage disequilibrium. And what's shown here on the bottom, this term selective sweep. A selective sweep is something where you have an advantageous locus that sweeps the population and becomes very common, and that can also happen with these neutral markers if they are linked physically close together to that adaptive marker. So that is a caveat there when you're thinking about just looking without any prior information and thinking about whether a marker is neutral or not. Sometimes it's a little bit more complicated if that neutral marker is located close to an adaptive one. So if we think back to that example we started this module with, where we had those two different studies with the intertidal snail, one of them said there were population differences and one of them said there were not. These two studies were actually done using different markers. So this study from 2010 used a neutral marker, and my study from 2016 used adaptive markers. And so this is just another example of how it's important to think about, based on the research question that you have to think about whether you want to use a neutral or an adaptive marker. And if you are interested in figuring out how individuals from different locations might be adapted differently to their environment, which is what I'm personally really interested in my research, then I had to make sure to use adaptive markers to ask my research question. So that's, again, an example of how choosing different types of markers can lead to different types of experimental results.